Hello, everyone. We're Team Petrified, and we did our presentation on defending against plant pathogens in recovering ecosystems. I'm Ruthvik.
I'm Caroline.
I'm Louie.
And I'm Antonio.
So pathogens have known to affect ecosystems, and this can be seen in agriculture when it comes to crops. So pathogens are estimated to cause around 12.5% of global crop losses. This can also be seen in recovering ecosystems. One example is the Big Sur wildfires, where a bloom of a specific pathogen happened, and this prevented certain vegetation to grow in that area.
Now that we know that plant pathogens exist, we can look at a possible solution. And that solution is microbes that actually exist in the soil. Now, these bacteria make up about 95% of all soil composition, meaning there's a giant reservoir of bacteria to pull from. Now, that's important because these microbes can be used for biocontrol. And biocontrol is using microbes to protect against pests, which in this case is going to be plant pathogens, to ultimately aid in the reduction of pathogen growth, as well as the reduction of usage of chemical pesticides and fungicides.
So given what we know about how wildfires change community compositions, we hypothesized that if these pathogens are a significant threat and they really do reduce the vegetation repopulation, then we should see observable properties in bacteria that we collect from an area of high and low plant growth after a wildfire.
And so basically, in order to assess this hypothesis, we sampled from an area in Getty View Park, which was victim to a fire back in 2017, which caused a lot of charring in the area. And basically, we wanted to focus mainly on an area that had a little bit of plant growth, but that had been affected by the fire. And we wanted to see if the bacterial community in this sample had any potential to protect against pathogens that existed in that area, whether it be bacterial or fungal. And so basically, we broke down our research project into two sides: cultivation dependent, where we worked with a lot inside of the lab, and we used a lot of the soil sample with antibiotics, fungal pathogens, and other things. And the cultivation independent part, where we basically used computer programming in order to basically assess the differences in areas that we cared about, mainly looking at areas of low plant growth as well as areas of high plant growth, and basically assessing the differences between the two in terms of bacteria that are present that could be beneficial to this pathogen problem.
And in order to know what bacteria was in our samples, we wanted to focus on the differences between these different areas, so the few plants and the areas with a lot of plants. And we wanted to specifically identify those bacteria and know if they could protect plants.
And so basically what we looked at was the alpha diversity of the bacteria. And basically what we looked at here was something using the Chow-1 method. And basically what that tells us is how unique the bacteria are within a certain sample. And so if you look in the top two rows, you'll see that these two samples correspond to areas that have few plants, basically the ones that we took samples from. And the bottom two rows were that of many plants, where other individuals have taken samples from. And basically when we used this Chow-1 metric, we found that there weren't really any differences in how unique the bacteria were in these two sample sites.
And for part of the diversity as well, we use a generalized unifact, which measures the similarity of bacteria between samples. And in this case, we have the samples from the area with many plants, and they're pretty close together. And then we also have samples from an area with few plants, and they're pretty far from each other, and they're actually in totally separate quadrants, which doesn't help us determine how similar the samples actually are.
And now that we know diversity and how diverse our samples are, we can actually look to identify specific bacteria in our samples. And we do that by isolating these things called biomarkers, which are special indicators that give information about bacteria that are present in an environment. And when we did that at the two different samples of areas of high plant density, as well as areas of low plant density, we found specific bacteria such as streptomyces, which is an antibiotic producing bacteria in the area of high plant density, and Pedobacter, which has antifungal production properties in the area of low plant density.
In addition to the biomarkers, we also utilized DNA sequencing technology in an attempt to identify our isolates further. And using a website known as Phylogeny FR, we were able to construct this phylogenetic tree which tells us the same information, that the bacteria that we found is related to Streptomyces and Pedobacter, and as was said prior, both of those are anti-pathogenic producing bacteria. And now that we know what bacteria is present in our samples, we wanted to see what specifically these bacteria were doing in these samples. So in order to do that, we observed the bacteria as we grew them in different exposures, and then we also wanted to see whether these behaviors are going to be useful at all in protecting against plant pathogens.
And one thing we can test for is these compounds called siderophores. Now, in the environment, especially in a post-wildfire setting, iron is a really important nutrient, but plants can't take up iron, so it's left up to competition between either the pathogens or the microbes. Now, siderophores are these pathogens that the microbes produce in order to help attract the iron into the cells of the bacteria and not the cells of the pathogens. And the way we tested this in lab was we grew the bacteria on a special color-changing media that changes color if siderophores are present, ultimately showing us that 9 of our bacteria, or our isolates, produce siderophores and 12 of our bacteria did not.
One of the other abilities that we can test for is the ability to defend against fungal pathogens, which is significant because fungi are among the most dominant causal agents of plant diseases. And what we found was that when we grew our bacteria in the presence of a plant fungus known as Fusarium oxysporum, 11 of the 21 isolates we tested were able to stop the Fusarium's growth on a Petri dish.
So we were also able to figure out antibiotic resistance within our bacteria. And from here, we grew bacteria in the presence of different antibiotics. And this is important because it helps us determine what specific bacteria are resistant to, and we can use that to kill off specific pathogens. So the picture that is circled in the blue circle shows that that bacteria is resistant to that antibiotic. And on the other side, you can see the average diameter of clearing per isolate for 2 specific antibiotics that we tested against.
So as for the experiences in the lab, one of the hardships that we had to deal with was basically the limited resources that we had for the class. Considering we were working with 3 other teams in a room, we were limited to the amount of biosafety cabinet time that we had to work with our isolates and our key assays. And so basically what we did to overcome this was to use office hours provided by our teaching assistant in order to basically knock out as many of the assays as we wanted to in order to get the results that we wanted.
We were also able to work with different teams and share different ideas and able to learn from each other. So if an experiment went wrong, we can get input from other people.
And the lab experience as a whole helped us expand our knowledge on how bacteria is able to interact with other bacteria present, as well as how it's able to interact with the environment that it's in.
And some of the experiences we gained, especially with things like antibiotics, can help us prepare for future careers in healthcare, where issues like antibiotic-resistant bacteria are becoming even bigger and bigger of a threat.
And with that, that is the end of our presentation, and we thank you all for watching.